did decide we're going to have the exam in class.
How many questions will be on the exam?
It'll be pretty similar to the previous one. Probably somewhere between 45 and 50 multiple choice, about 4 short answer questions. Oh, you got one. Okay, does everybody have a handout? Everybody got one? Okay. Now, you will have to do something like this on the exam tomorrow, okay? So we're going to go through how you do this. And what we're going to do is we're going to do central dogma. We're going to go from DNA to RNA to protein, okay? Amino acid sequence. So what I'm going to start, I didn't start it. Okay. So what we're going to have to do is go DNA to RNA to protein. And you have a handout. If you are not in class and you're listening to this, just print this out. It's at the top of the page here, and then work through it with us. So this is the DNA sequence. How can you tell by looking that that's the DNA sequence?
There's T's.
What?
There's the thymine.
There's thymine in it, right? So that tells you that it's DNA and not RNA, okay? How many bases are in a codon? 3. Okay, so the first thing I recommend that you do, this is the way I would approach this problem if I were doing it. It's hard to remember a sequence of letters. Most of us have limited ability. Our eyes start losing focus when you're looking at a whole string like this. So the very first thing I would do is go through and put a peck mark of some sort every 3 bases. That will make it much easier to keep up with where you are. So go through and every 3rd base, put a little slash. Okay. Now, how many sets are there? There's 11. So every step you go, make sure you have 11. Because the next easiest thing to do is to skip one. Skip a whole triplet, okay? So mark them off every 3, and then keep track and make sure every time you do this that you end up with 11, okay? So the next step will be making the RNA from the DNA template. Now, are we turning DNA into RNA? No, the DNA is still going to be there after we do, what's this process going to be called, DNA to RNA? Transcription. So after we do transcription, DNA is still there. Okay, it's going to get zippered back up, it's ready to use again. So it's just a template for the RNA. So what's going to match up to TAC? What would be the first RNA triplet? AUG. Why wouldn't it be ATG? Because it's going to be RNA. RNA does not contain T. So our very first code is going to be AUG. Then put another slash mark. What's the second one going to be? GCA. Okay, so you write GCA. I would try to write small enough to keep it kind of in line. one going to be? ACG. Don't do this too fast, because especially with the C's and G's, it's really easy to get them mixed up. Okay? What's the next one going to be? UAC, right? Okay, now finish that row. And when you're finished, count to make sure you have 11 codons. What's going on back there? Put your Instagrams away or whatever you're doing. Okay, has everybody got it? All right, so your messenger RNA should look like this. So check to make sure you have it correct. Anybody can make a mistake, okay? And in fact, that's what a mutation is. It's just a mistake. Okay, everybody got it? All right, so what's the next step? We've done transcription now. Translation. And in translation, are we turning RNA into protein or into amino acids? No, we're just coding for them. And then that messenger RNA can get used over and over and over and over again. Okay? So, AUG is our first codon. So, AA stands not for Alcoholics Anonymous here, it stands for amino acid. Okay? So, our first codon is AUG. Usually we can remember 3 letters. Okay? Most of us can remember 3 letters fairly easily. So, we go to the genetic code and we find AUG. It stands for MET, or methionine. So you would write on your sheet, you would write MET. Do not try to write out methionine.
So are we doing a dash or between lines?
I'm sorry?
Are we doing the dash or between lines?
It doesn't matter. Here I use 2 little dashes. It really doesn't matter. I am sometimes inconsistent. Okay? Now what's the next codon? GCA. So we go here and we go G, here's G, C, A. And what does that code for? Alanine, ALA. Believe it or not, there's actually 1-letter codes for all the amino acids, which is a real pain because some of them start with the same letter, so they have weird 1-letter abbreviations. But again, if you use it all the time, you will know that. You will know what those codes are, since we don't use it all the time. You need to get a new mask. If your mask will not stay up over your nose, get you a new mask. Okay? I've got some up here if you want one. But you have got, your mask does not work. Most of the germs come out of your nose, not out of your mouth. Okay, now this is where I used to tell people you had to memorize the genetic code, and some people would dutifully write that down, and other people would look like they're in panic. You don't have to memorize the genetic code. All right, what is the next codon? ACG. So we go A, C, G, and that stands for 3-indenous. Some of you in the back are having a hard time reading this, but you'll have one in your hand. THR, the 3-indenous. Okay. What's the next one? UAC. So go here and we say UAC is tyrosine, TYR. Okay, so go ahead and finish that and write all the amino acids for your codes. You have them on your paper. So. Anybody having trouble using the code? Again, I know if you're in that, you probably have trouble. Especially, eyes are not as good as they could be. Okay, so here's what it should be: methionine, alanine, threonine, tryptophan, proline, lysine, histidine, threonine, serine, glutamate or glutamic acid, and stop. I will not deliberately put a stop codon in the middle of a sequence. If you're doing a problem and you hit a stop codon in the middle of a sequence, either I've made a mistake or you've made a mistake. Okay? I will not do that on purpose. Do you have 11 entities up there? Yes. Okay, the stop counts as one of those 11. Okay? Questions about doing this? It's really pretty straightforward. You just have to remember how to do it. Okay? Now, mutations. Mutations are changes in the DNA sequence. That's the definition of a mutation. It's a change in the DNA sequence. For example, in normal hemoglobin, at a particular point, it's fairly early in the sequence, the DNA should be CTT, and the messenger RNA should then be GAA, which codes for glutamic acid. But in the mutant hemoglobin, instead of being CTT, it's CAT. And the messenger RNA is GUA, which codes for valence. In this particular case, it's a devastating mutation because it makes, I don't know why it keeps cutting out. It makes the protein not function properly. There are a couple of different kinds of mutations we're going to talk about, and then we're going to do examples on your sheet, okay? There is what we call there are point mutations, and one of those is called a base substitution. And that's what was just described in the sickle cell anemia. In this case, in this particular one, a serine replaces a glycine. So there's lots of different mutations that can take place. And so a substitution mutation can result in a change in the amino acids. So let's try this. Let's pretend that the whole sequence is the same except for the second codon. Okay, we're just going to look at a mutation in the second codon. So if the original sequence is TAC-CGT, and our original messenger RNA is AUG-GCA, and our amino acids would then be methionine and alanine, right? That's what we just did. So that's pulling that down again, okay? Now, mutation number 1, our DNA is TAC-CGG. What have we changed? We changed this thymine to a guanine, okay? So what would the messenger RNA be? AUG GCC. Well, we already know that AUG codes for methionine, okay? So you can go ahead and write that down. What is CGG going to code for? C or CG, where is it? G, where's my G?
Bottom.
Bottom, okay, GCC. Okay, what does GCC code for? Alanine. So what happens with this particular mutation? What is the result of that mutation?
No change.
No change. So this is an example of how a mutation can have no effect. So this mutation has no effect at all. And if you look back at the genetic code, it's one of those cases where it doesn't matter what the third letter is, right? It's all the same. We sometimes call that wobble. Okay? Let's look at the second mutation. This time it's TAC-CAT. What did we change this time? We changed a guanine to a adenine. Okay? So what's the messenger RNA going to be? AUG-GUA. What does AUG code for? Methionine. It's always the first one. What does GUA code for? We look for GUA. It codes for valine, V-A-L, valine. Okay? So what I've done here is drawn the structure. This is the structure of alanine. This is the structure of valine. Now, I don't expect you at this point to be able to look at those and know anything about them, but I'm going to point out what the difference is, okay? If you look at the top, this part I'm drawing a circle around right here, it's the same on both amino acids. Let me do it here so people can see it on the tape. Okay, so if I draw a circle around here, around this part, and around this part, they look the same, right? It's this bottom part, which is the R group, which is different. Here it's just a CH3, which is a methyl group. Down here, it's actually a butyl group, or an isobutyl group. Now, that makes this one bigger than that one, right? But it would be like natural gas versus butane in a lighter, okay? What do they have in common? What does natural gas or methane have in common with butane?
Flammable.
They're both flammable. They're both non-polar. They wouldn't mix with water. So chemically, they are very similar. Okay? You're going to have to trust me. If you're not big on chemistry, you're going to have to trust me on that one. But chemically, they behave in a very similar manner. One's just bigger than the other. Okay? So chemically, they are going to be different, but they have a similar structure. They are both, for example, non-polar amino acids. So they're probably not going to have a huge effect on the protein. It's still going to fold the same way. It's not going to form the same kind of bonds in its tertiary structure. It might work a little faster or a little slower than the original protein, but it's still going to work. Protein's still going to work. So it does cause a difference. It does cause a slight difference or a minor difference. Could be beneficial, could be detrimental, but it's probably not going to be devastating. Okay? Third mutation. This time we go from TAC to CTT. What is the difference this time? We've changed the guanine to a thymine. So what's going to happen to our messenger RNA? GAA codes for. So we go GAA. What does GAA code for? Glutamic acid. Here's the structure of glutamic acid. Again, if you look at the top, like the top two rows here, it's exactly the same. That's what makes it an amino acid. Okay? But this one, the bottom is just that non-polar methane group. But here the bottom contains a charge. It contains an extra acid group, which happens to be at most normal pHs is charged. It has a negative charge. It is not non-polar. It is actually like substituting salt in a recipe for sugar, a little bit. Or substituting salt for oil, that might be better. Because the other one was non-polar like oil. And so it would be substituting salt for oil in a recipe. Would that work in a recipe? No, not only would it taste bad, unless you're a huge salt monster like my husband, he'd probably love it. But it wouldn't rise properly. It wouldn't work. It wouldn't cook correctly. The chemistry doesn't work. Besides, the taste would be awful. Okay? So this time the change in the genetic code means we've replaced a non-polar amino acid with a charged amino acid, like replacing sugar with salt or oil with salt in a recipe. It won't work. So, what I want you to get at, and let's look at this mutation and then we'll have a summary here in a second. There are also mutations that we call insertions or deletions. And in the case of an insertion or deletion, you actually take away or you put in an extra base. So the example I sometimes use that's easy to see let's say you had a sequence that looked like this: cat, 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 cat. Turn this over that way. Not sure you can see anything. Okay, cat, 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 cat. All right, if we go every 3, it looks like this, right? What if I took out this letter A? What would happen if I did a deletion where I took out that letter A? The first one would be still cat. What would the second one now be? C, T, C, and then ATC, then ATC, then ATC, and so on. Okay? So this is called a frame shift mutation because you've changed the reading frame. You're not seeing the same group of, the same cluster of 3 anymore. So a frame shift mutation can be exceedingly bad because it not only changes one amino acid, it changes every amino acid from that point forward. Okay? So that would be a devastating, probably a lethal kind of mutation for that protein. It would make that protein non-functional. So here's an example. So if this is a sequence, oh, I guess I did put it up there. Okay, I didn't realize I'd done that. So it should be all valines, and now we take one out, and it becomes, in this particular example, it goes valine, 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 glutamine, stop. Okay, I forgot I stuck that in there. That's a pretty devastating mutation, and the protein won't work. So the loss of a single nucleotide can completely destroy the function. Now, This you need to know for the exam. This slide is extraordinarily important. I need to switch this back. Okay. I'm going to ask you this question on the exam. It's on Canvas. It's all on Canvas. Okay. How could a mutation have no effect, a minimal or mild effect, or a dramatic effect? If a mutation causes no change in the amino acid sequence or is in part of the DNA that is not used for transcription, mutation will have no effect. Because we don't use all our DNA. If a substitution mutation causes a change of the amino acid sequence, but substitutes an amino acid very chemically similar to the original amino acid, there may be a slight effect or a minor effect. If a substitution mutation causes the change of the amino acid sequence and substitutes a chemically different amino acid, or if a deletion or insertion changes the reading frame, the effects can be devastating. So this is the answer to the question, how can a mutation have no effect, a minor effect, a major effect? You don't have to write all this down. It is in the Canvas PowerPoint. Okay? The question is, do you understand what that says? Does it make sense to you? Anybody have a question about it? A mutagen, I think we talked about this, didn't we? All right. So I need to go to okay. So now we're going to go back. Okay. This is where we were Tuesday, correct? And we were talking about polio, correct?
Yes.
And we were just starting to talk about how they developed the vaccine, right? Okay, so in the 1930s and 40s, there were a couple of different, there were several different research labs working on a vaccine for polio. And one of them was Jonas Salk, who you've probably heard of. He's the one who won the Nobel Prize. So what he developed, first of all, isolating the virus is very difficult. It was not an easy process to develop a virus, to isolate the virus with the technology they have. It's still not simple it's not something we can do in a freshman, we can do bacteria in a freshman biology lab. We can't really do viruses very much in a freshman biology lab with the technology we have. Now, when you get a little further along, you can, but it's a little tough to do. So anyway, he had to first isolate the virus and then come up with a way to spark the immune system to fight the virus. So what he developed, he took the polio virus and killed it and then came up with a vaccine that was reasonably effective. I don't remember the percentage, but let's say it was about 80% effective. It was an injected vaccine. It was a shot, okay? And I believe they started giving it in the early 50s, maybe late 40s. And I think I told you, parents were terrified of polio because it could cause death. It could cause a child to end up with what they call an iron lung. Because what polio does is paralyze skeletal muscle. And the muscles you use to breathe are skeletal muscles, not cardiac muscle or smooth muscle. You have control over your breathing. Now, most of the time you don't think about it. We have processes that just kind of make you breathe, okay? But you can hold your breath. You can adjust your breathing if you choose to, right? That's because it's skeletal muscle. So if the breathing muscles get paralyzed, what they used an iron lung for was to change pressure. And what would happen would be it would oscillate between increasing pressure in this iron lung, which would push the chest cavity in and cause you to breathe out, and then it would lower pressure, which would cause the chest cavity to expand and breathe in. That's how breathing basically works. You expand and contract your chest cavity, okay? So the first vaccine was reasonably effective, but there were still a lot of cases of polio because it's highly contagious and can be deadly. So at the same time, I think his name was John, but I'm not sure, Sabin, another researcher, instead of using a killed virus, he used what's called an attenuated virus. In other words, he took a polio virus, exposed it, I think, to radiation. I'm not absolutely sure about that, but I think he exposed it to radiation or something. And cultured out an attenuated virus that didn't cause polio, but could still spark the immune system to respond. And this became an oral vaccine. And you all probably had the injected one, but my kids had the oral one. The oral one works better because it turns out that polio is actually what we call an enteric virus. In other words, it attacks the body through the digestive system. So having an oral vaccine means it stimulates, a lot of your immune system is within your GI tract, and so it stimulates the GI tract. And it turns out, it's kind of gross, but if a baby gets a vaccine, whoever changes their diaper gets a little bit of dose of the vaccine, okay? It just works that way. It's kind of like a booster. And so when the World Health Organization and the CDC and all the people who make decisions found that the oral vaccine works better, it works like 7% of the time. So it's much better from a public health perspective to use the oral vaccine. Here's the kicker. 1 in 10 million children who get the oral vaccine will get polio from the vaccine. It's a really tiny percentage, but that's still there because somehow the attenuated virus becomes active in that child's body. Somehow the immune system turns it back on. Well, that's a really bad thing. So Salk always argued against using the Sabin vaccine, but the Sabin people said, but it protects more people. And so when you're looking at things from a public health perspective, the more people you protect, the better it is, right? So this was an argument for decades. In the 1990s, Dana was born in 89 and David was born in 92. Somewhere about 1990 in the United States, they finally switched back from using the oral vaccine to using the injected vaccine because the levels of polio were so low. Once you get down to a certain level, then the less effective vaccine is okay because your population is not exposed to polio very often, okay? But in developing countries, you still need to use the oral vaccine. They probably should have switched back to the injected vaccine sooner. They probably should have done it maybe in 1980, but they were afraid to. So there's a certain number of children who got polio between that time frame. Now, when you talk to people who are anti-vaxxers, may include some of you, or your parents, when you talk to people who are anti-vaxxers, one of the things they bring up is the fact that the United States has a vaccine fund that is for people who are injured by vaccines. That fund exists because of the polio vaccine, because of this one case. All right? Anybody whose child has a vaccine and they make of any sort and they make a claim, they automatically pay it. Okay? There is no science that goes into it. I mean, if they get a doctor or something to say, yes, this child had a vaccine and this happened to them, they pretty much pay it out. They don't argue, they don't litigate it for the most part. And so the anti-vaxxers use that as proof that vaccines are dangerous. That's not how it works, okay? So it was developed simply because they wanted people to have the polio vaccine. We went from a high percentage, I don't know about high, but a reasonable percentage of people who had polio to virtually nobody having polio in a really brief time. And this was such a big public health issue that I remember standing in line at the local junior high school. We had to go 3 Sunday afternoons a few months apart to get the oral vaccine. I probably had the injected one as an infant. But I was probably 3 or 4 years old, and I remember standing in line. Every person in the community, there was no argument. There was no, this vaccine might hurt my child. Everybody went. And you got a little squirt in your mouth. No, then it was a sugar cube. My kids got a little squirt in the mouth. Like a little pouch that pediatrician just squirted in their mouth. A little pink liquid. But for us, it was a sugar cube. So, that's the story of the polio vaccine and the anti-vax. That's where that fund came from. And that fund is funded by the vaccine makers. And the government probably kicks in some money as well. So that is the story of the polio vaccine. It was the second disease that the World Health Organization targeted for eradication. Got rid of smallpox through vaccination. Almost got there with polio until AIDS hit. And there were wars in Africa and the Middle East. And dictators, I think I already told you, dictators decided that the big bad Americans were infecting people with AIDS through the polio vaccine. And so they rejected it. Polio made a big comeback. Let me tell you, my older brother, who's 3 years older than me, had kids in his class that had polio. I don't know anybody younger than my brother who had polio. Nobody in my class had polio. That's how big a difference that oral vaccine made. In 3 years, virtually wiped it out, at least in suburban America. And I know a few people who have what we now call post-polio syndrome, where they had polio. It probably permanently affected their muscles, but your body recruits other muscles to take the place, but then they fatigue over time when that was not their original job. Anybody's interested in physical therapy, you'll learn about that. But if muscles are destroyed, other muscles will come in and kind of take over the function. But since it's not their original function, they tend to fatigue over a lifespan. So we had a next door neighbor who had polio, really good friend of mine. Thinks she probably had it because her parents were weird. They didn't take them to the doctor ever. And her dad was in the service. I mean, they had free health care, and her mom still did. She said she remembers waking up and she couldn't walk. And her mom didn't take her to the doctor. I know, and it's like, she's like, I don't know what. They had a bunch of kids. I mean, she's got like 8 brothers and sisters. But, you know, if your kid wakes up and can't walk, I mean, it's just, she's like, I don't know what she was thinking. But anyway, so she probably had polio. And it varied. You know, some people had a very severe case, some people had a very mild case of polio. So that's kind of the story on polio. Hepatitis A, they now have a vaccine. That's the one that made a big comeback a couple years ago. In fact, I think I got the first vaccine for it. That's the one that can be spread by restaurant workers, typically. If you have not been vaccinated for Hep A, you probably should be. Okay? And there's other enteric viruses. Okay, coronavirus. We get to hit coronavirus. We used to really just sort of smooth over coronavirus. It usually causes a common cold. Most coronaviruses. Most people have probably had a coronavirus in their life. It also causes something called SARS, and I may have a picture of, no, I think that was this picture. This is a picture when SARS was a problem. But so SARS is severe acute respiratory syndrome. They thought it was going to be a pandemic, and it may have technically met the definition. But the world reacted very quickly to SARS and kept it contained, and it did not become a worldwide problem. It was in the, I think in 2012, sometime between 2000 and 2012 or something like that is when SARS happened. Okay, certainly I remember. There's another one called MERS, which was Middle East Respiratory Syndrome, which is another coronavirus. And now we have COVID-19. What is the problem with COVID-19? What are some of the problems? A lot of problems. What's the problem? What? You can spread it when you're asymptomatic or pre-symptomatic. Now, I had somebody at church, drives me crazy, but asymptomatic, you can be asymptomatic and have the virus, okay? And you can spread it, turns out. We didn't know that initially, but it turns out you can. You can also, but then she was tested and then she showed up for surgery, and on her chart, when she looked at her chart, it said asymptomatic, and she got all freaked out because she thought that meant she had it, but she was asymptomatic. All asymptomatic means is you don't have symptoms. That means the day she showed up for surgery, she didn't have a fever, she didn't have a cough. It doesn't mean she was positive and asymptomatic. It just meant she was asymptomatic, okay? So you can be positive and asymptomatic or negative and asymptomatic, okay? But it just means you don't have any symptoms. You can also be pre-symptomatic. That means you can have it and you're going to get sick, but today you don't. Today you're not sick, okay? You can still spread it. What's another problem with SARS, with COVID-19? Say that again?
I'm heard it's like not, it doesn't like go away very quickly when it's like in the atmosphere. Like it takes like a few hours for like if it's in the air.
You know, yeah, but you have to get a certain dosage of it. In fact, they think one of the reasons that wearing a mask, you know, initially they thought wearing a mask only helped protect other people. It wasn't helping you protect yourself. It turns out wearing a mask does help the mask wearer, and they think it's because it reduces any load, it reduces how much, and it takes a certain number of viruses inhaled, essentially, to cause any problems. Your immune system can handle a few. You have to have a certain dose before you have a problem. Yeah, it might hang around in the air for a little while, but it mostly is attached to water droplets, and water droplets fall fast. One of the problems with COVID is that it is what we call a novel coronavirus. And that means we haven't been exposed to it before. It's new. It moved from animals to humans. And we don't have any innate immunity necessarily to it because we haven't been exposed to it before. So that becomes problematic. Developing vaccines takes time. It's a problem. Rushing the vaccine to market is not a good idea. The pharmaceutical companies came out yesterday or day before and said absolutely they will not bring a vaccine to market that has not been through all the testing. Now, they are doing it rapidly. That doesn't mean it's bad. What they're doing is not bad. They're still going through all the trials. It's just usually this takes a lot longer because it's not in between everything.
Isn't there supposed to be like a vaccine like November 1st?
No, that's politics. And that's another problem with the coronavirus. What has happened with the coronavirus is it has become a political issue instead of a medical issue. When you look at who will wear a mask and who refuses to properly wear a mask, it almost falls completely along political lines. And that is absurd. Absolutely absurd. We should be following what the medical personnel and the scientific personnel tell us, not what the politicians think. If they are following what the public health and the doctors are saying, then we should listen. If they are not following what the public health and medical researchers say, they don't have any special knowledge because they're politicians. I mean, having a business degree does not make you a medical expert. Okay? Doesn't. Sorry. Should not be a political issue. Do masks work?
Yes.
How do we know masks work?
Testing is cases that went down.
We tested them, what?
Cases that also went down since people wear masks.
Yeah, if we, again, this person at church who drives me insane, she comes to me and asks me all these questions. And she's like, well, why are our cases still in the 700s every day if we're wearing masks? It's like, well, because you have this spike, and now you have all these people who are spreading it. I said, when you only had 200 people a day spreading it, you didn't have as many cases as you have when you have 700 people a day spreading it. I mean, that only makes sense. They're not going to start coming down immediately because you still got a lot of people spreading it. It's going to take a while. It has a high R naught. Have you guys heard of R naught? That's how many people on average get it from one infected person. For the flu, it's actually less than 1. For COVID, it's somewhere between 2 and 3. And that makes your exponential growth much higher. When it's less than 1, like it is for the flu in general, it makes it not, I'm not saying the flu is not a huge problem, it is. But COVID has a higher R naught. It also has a higher death rate. And people who say X number of people die from the flu every year. Yeah, and those are the same people who are not getting vaccinated, which really irritates me. But yeah, people die from the flu. And yeah, it's the same kind of people in general who die from the flu that die from COVID. It's people who are immunocompromised. It's the elderly. But it's still a much smaller percentage, like orders of magnitude smaller percentage.
So when the flu first came out, like before we knew it, was it kind of like Corona where we didn't really know how to...
Say that again.
Like when the flu came out, like before we actually knew what it was it like Corona where it was like very severe?
Well, of course, we didn't have vaccines or anything. I mean, the flu has been around a long time. Humans have been dealing with influenza for a really long time. If you're interested, I heard a really cool podcast, I think it was a podcast, may have been an article I read, about the origins of influenza. It was really cool. But it's been around over 1,000 years at least. We only really recognize it as influenza as technology got to the point where we could isolate it and actually look at the virus. So, coronavirus, big problem. I just found out that my niece and nephew have it. She's a social worker in a hospital. That's where she got it. She gave it to her husband, who's an attorney. So they're about 30, 31. And they've been pretty sick. Not go to the hospital sick, but reasonably. I mean, lost their sense of taste and smell, lots of congestion, fever, body aches, fatigue, like really slammed like a bad flu.
Would you have corona? Can't you have it for more than 2 weeks? Or if you
You have the effects of coronavirus for more than 2 weeks. I have a cousin, she's actually my third cousin's wife, who's been in the ICU since the middle of May with corona. And she doesn't have an active coronavirus infection anymore. She's in ICU because it damaged her organs. So she's still on oxygen. She's, let's see, she's got some sort, I don't know if it's just from being in the hospital or what, but I know she's had to have several abdominal surgeries for infection, draining infections and stuff like that. They actually had to put her on what's called ECMO, which is extracorporeal something, which is like being on a heart-lung machine, basically. They take your blood out and oxygenate it outside the body instead of, to give her lungs a rest. My guess is she will need a lung transplant or she will be on oxygen forever. She's my age. She's like a year younger than me, had no pre-existing conditions. My cousin said she was the healthiest one in the family.
Why do people say, like, don't take ibuprofen with, like, in case you get the coronavirus?
Yeah, I can't remember what it is. It has to do with, it's probably because it's an anti-inflammatory. And so initially you want that kind of response. It's kind of a delicate balance because steroids, corticosteroids help, but not initially. Like if you've had bronchitis, they a lot of times will give you some prednisone or something. And it's kind of you have to be careful because those drugs decrease your immune response, but they also open your airways. So it's a delicate balance as to when you want the anti-inflammatory and when the inflammatory response is fighting the disease. So I think that's the issue.
Okay, this is something I heard about the experience after. I heard that there are certain blood types that they're figuring out now that
Yeah, they seem to think people with type A blood seem to have a worse case than people with type O blood. So I give my husband a hard time because I have O and he has A.
because we would, like, over the summer we went to the beach with, like, this family and they shared Alka-Seltzer or whatever, and we were all, like, in close prayers of each other, and then we got back.
That was not a good idea.
Right, well, we didn't leave anywhere. Like, we didn't go eat or anything. We just stayed in the house, at the beach house, and just went on the beach and stuff. And then, but we came back, and I noticed, like, my parents didn't tell me at first, but I guess the people that lived with us came back, and a day later, like, showed symptoms and tested positive. And so, like, my parents, like, stopped working for a few days, so, like, oh, we're just taking off to get back together, like, after going on vacation. And they started, like, I noticed they were, like, gone and, like, doing stuff, so they went and got tested. And they made me, well, sister tested off, came back negative, and then we went and got tested again, came back negative again. But we somehow didn't get it, and they all three, the other family, all three got it somehow. We, like, lived with them, like,
There's no telling.
it was really strange.
Well, for as long as you're positive for your infections,
They really don't know. You are less infectious after a few days. And that's, some of this stuff is just by experience. This seems to be what's happening. And so, they make, I think it's funny, if you're positive, you have to isolate for 10 days, I think it is. And if you're exposed, you have to isolate, you have to quarantine for 14 days. So it's worse to be exposed than it is to have it.
Okay, so I saw an article saying that your dog can get it. That's not true, is it?
Well, pets, you know, think about how many people who have had COVID have pets. And we know of about 5 cases of animals having it.
Yeah.
So it's extraordinarily rare.
Okay.
Don't kiss your dog in the face, okay? I mean, that's gross anyway.
What if pets don't, I know dogs and cats have like a vaccine for corona, it's not COVID-19. Do you think that holds it?
I don't know. When we had a dog, I don't remember getting a coronavirus vaccine, but that's been a few years.
Why is it that, like, because there are other coronavirus vaccines, and I think there's one for cows specifically, why is it that scientists can't take those vaccines and, like...
I'm sure they're working on it. I'm sure that's one of the things they're doing. You know, scientists are smart people. They're aware of these things. So. I mean, with my cousins, they're both physicians. You know, my cousin is he was an endocrinologist. He's retired now. He made enough money he could retire when he was like 60. But she is an infectious disease physician. And she was running the COVID response in the hospital in Bowling Green. And she didn't get it from the hospital. She got it from her mother-in-law, who's my second cousin, my mother's first cousin, who's 90 now. And she got it from a caregiver. My elderly cousin was in the hospital for 5 days. 5 days, not in ICU, just in the hospital, had to go to rehab. Yet, my cousin that's my age has been in ICU for June, July, August, September, now four solid months in ICU, and she was healthy.
Is SARS like more, like, is corona more severe than SARS?
No, SARS is worse. Turns out SARS has a higher death rate than COVID-19, I think. I think it was more like 5 to 8% fatality. It was pretty high.
Yeah, because in like December, my dad, he got really sick, and they said it was SARS, but it might have been corona because like they gave him like medication for SARS that like they had for that, and like it didn't work out well for him.
Yeah, to my knowledge, SARS is not. I mean, the word SARS just stands for severe acute respiratory syndrome. So if you have an acute respiratory syndrome, you can call it SARS, but that doesn't mean it's caused by the coronavirus. Okay? All right. Yellow fever is caused by a flavivirus, so is West Nile and hepatitis C. Hepatitis C you've probably been vaccinated for. They started vaccinating children back in about 1994. I could see the writing on the wall that they were going to start requiring a Hep C vaccination, so I went ahead and got my kids vaccinated. Their peers had to get vaccinated when they went to middle school, and they had already had it, so they were really glad. It's a series of 3 shots, I think.
What's the difference between like hepatitis C and hepatitis A?
They're just different. They all affect the liver, which is why they're called hepatitis, but they're very different viruses. They're different categories of viruses. Hep C is a bloodborne virus. It's contracted the same way AIDS is, but AIDS, Hep C is a lot more contagious than AIDS is. It's easier to get Hep C than it is to get AIDS. That doesn't mean it's hard to get AIDS, I just mean it. People get Hep C easier. If somebody's both Hep C and HIV positive, their partner's much more likely to get Hep C than to get AIDS. All right, what do yellow fever and West Nile have in common? Anybody know? Mosquitoes, they're both spread by mosquitoes. So, West Nile is not that big a deal in most humans, it's huge in horses. It's a big problem with horses. Yellow fever, more people died. People don't realize, it drives me crazy when they don't want to get vaccination. More people died in the Spanish-American War from yellow fever and malaria than died in the war. More people died in the Civil War from measles, mumps, and seems like something else, probably the flu, than died in battle, from battle wounds and infections from battle wounds. It's incredible. Togavirus causes rubella and equine encephalitis. Rubella, I want to talk about rubella. When I was a child, rubella was called the German measles, the 3-day measles. They caused they were just one of those, remember I said this disease, it was one of those rashy fever diseases that kids got. The problem is, if a pregnant woman gets rubella, the baby, depending what part of pregnancy she's in, the baby can have severe birth defects. Severe. And so this often happened to women who had several children. The first child goes off to kindergarten, comes home with the German measles, gives it to the pregnant mom, and then the baby has severe birth defects. It was one of the leading causes of blindness and deafness, particularly deafness in children. My friend that I told you probably had polio was a teacher for the blind and deaf for many, many years. And when she found out people had stopped vaccinating, because that's the R in the MMR, she just went ballistic. Because when she first started teaching, most of the children who were deaf because the mothers had rubella. Now we have a vaccine. There shouldn't be any more children deaf from rubella. Shouldn't happen. They are so concerned about it. I probably had rubella as a child. But they are so concerned. When I was supposed to get married when I was 22. Everywhere I went, like get the marriage license and get my gun, everybody wanted to vaccinate me against rubella. That's how big a deal it was. I've probably been vaccinated for rubella about 4 times. Okay? Because that's how worried they are. It causes deafness, it causes mental retardation, it causes limb abnormalities. There's a little girl, when I was little, my best friend's mother had a baby, and she had rubella. Baby was born with an ankle abnormality. She was deaf, profoundly deaf, and she was developmentally delayed. It was really sad. Filovirus, Ebola. Again, it's one of those emerging viruses we have no immunity, virtually no immunity for. Interestingly, and this is why information is important and understanding science is important. I remember getting so angry listening to some guy on the radio who said if the ozone in the air you breathe, ozone is a pollutant. If you have asthma or something, breathing ozone is bad, okay? But ozone in the upper atmosphere helps block out UV radiation, okay? So it's good one place, bad another place, okay? Ebola is different from the flu. It's different from COVID-19. It's different from a lot of viruses because it's not contagious in the early stages. There was a guy in Texas. He came back from one of the countries that had Ebola. He was living in an apartment with a bunch of people, immigrant family, unfortunately. There were like 10 people living in an apartment. He went to the emergency room because he was sick. They did not diagnose him with Ebola, but he had some of the symptoms. Okay? He went back, they sent him back home. He got sicker and sicker and sicker. The family called an ambulance. He was actively throwing up. The ambulance workers, his whole family, people in his apartment building, people in the ER, until they realized he had Ebola. All those people were exposed to Ebola. You know how many of those people got it? 0. Not one person. But when he was in ICU and they were using all the precautions, but something went wrong and he was dying, 2 nurses got it. 2 nurses who took care of him in his last days got the virus. Because Ebola is not generally contagious until the person is pretty much dying. And it spread in Africa so much because the culture in Africa, you take care of your sick, you take care of people when they die, you wash the bodies and you do all this. And so people were being exposed to it when it was most contagious. Okay? So Ebola is different. You have to follow the science. So you don't freak out because there was a doctor who came back who developed Ebola and he was walking around Manhattan before he had any symptoms. Okay? He didn't spread it to anybody. Ebola is not contagious then. COVID is. It's not a political statement. It's science. Okay? Influenza. We have to talk about influenza. Get your damn flu shot. Okay? Who dies from the flu? Do college students die from the flu?
No.
Very seldom. Once in a while. Once in a while it happens. But do you have old people in your life that you love?
Yes.
You know what? They die from the flu. You know what? They get the flu vaccine. But you know what? Their immune systems don't work as well. The flu vaccine does not work as well for the elderly. So when you go see Grandma over Christmas, hopefully, we get to go see Grandma over Christmas, and you see Great Aunt Susie, and you see the guy at church who used to give you gum when you were a little kid, those people, you can take the flu to them. And that's really bad, and you might not ever get sick. You can give it to me. I get the flu shot. But we don't mostly have substitutes. You know? Once in a while we'll cover each other's lab or lecture, but we don't call a substitute teacher to come in. So if I get sick, for the most part, my classes don't meet. Okay? So I don't want to get the flu from you guys, and you're living in a petri dish. So get your flu shot. You cannot get the flu from a flu shot. I don't care what your Aunt Susie says. It is a killed virus. There is no way for a killed virus. Nobody got polio from the Salk vaccine. They got polio from the Sabin vaccine, which was an attenuated virus. If you are super concerned, do not get the nasal one. The nasal one is an attenuated virus. But the injected flu vaccine is a killed virus. Yes, you may get a few symptoms because most of the symptoms of the flu are from your immune system. Fever, shakes, you know, things that your immune, snotty nose, that's your immune system responding. Don't get your flu shot the day before the big game, only there's no big games now, so you don't have to worry about that so much. Don't get your flu shot the day before the big test, in case you have a little bit of a reaction to it. Yes, you can still get the flu when you get the flu vaccine, but you won't get it as bad. They've run the numbers. You don't get it as bad if you have the vaccine.
Oh, I was just gonna ask, isn't there like a spike now because more people aren't getting vaccinated?
I can't.
Isn't there a spike in influenza because more people will get vaccinated?
Yes. People are weird. I don't know what the problem is. It's become political. It's science, people. All right. Rabies, I got more stories to tell you. We'll pick up on this maybe after the exam tomorrow. Okay? I will see you tomorrow.